Mr. Bondi here with our next biology lesson, this time on cell membranes and on cell transport. In this image here, you can see Elodea. It's an aquatic plant, and we're looking at it magnified 400 times, so under high power of our microscopes. You can clearly see the outline or the boundaries of each of the cells here, and there's probably something around 20 or so cells in this image here. But what I've done is I've highlighted one cell specifically, and inside of that cell, you can see a bunch of green spheres. Those are all the chloroplasts, the structures that are responsible for doing photosynthesis. But really what I want you to focus on is the line that makes up the outside of it. That's the cell wall. Now, that's not the main feature that we're going to focus on today, but what we're going to look at is something called a cell membrane. And so in this animation here, what you can see is an orange sphere representing a cell with a little slice taken out of it so you can see some of the interior components of it. You can see the nucleus and some organelles and maybe some proteins floating around inside of it. But what's key is when we zoom in on just one little piece of that membrane, and we can see that zoom in over here on the right-hand side. And you see these structures right here, which look like a bunch of orange spheres with these little red things dangling off of them. These are phospholipids, which make up the main structure of the cell membrane. And really, our cell membranes are used for three things. The first of which is that they're used as a barrier. They're there to keep the outside stuff out and the inside stuff in for a cell. They're also used as a way to move materials in and out of a cell so that the cell can maintain homeostasis. And then finally, they can be used as a way to identify the type of cell by the things that they have on the outside of them called antigens. So here we have another cross-section of a cell membrane with all of its parts. And as you take a look at this, there's a lot to see, but the main things to take note of are all of these red spheres with like the orangish yellow tails hanging off of them. There's by far the most of those. And those are phospholipids. Here's an individual look at a single phospholipid, right? And it has two major parts that we can see. The first part I've bracketed here is a polar head. And I say polar because the bonds inside of this part of it have polar covalent bonds. And if we remember, polar covalent bonds are the types of bonds that you find inside of water. So this portion of the phospholipid likes water. The lower portion here, which actually has two fatty acid chains, one saturated fat and one unsaturated fat, is nonpolar because it has nonpolar bonds. So this portion of the phospholipid would actually dislike water. It's really important that we take a note that this phospholipid has parts that like water and parts that don't like water. If we take a look at how the phospholipids arrange themselves, they do so in what's called a bilayer, bi in this case meaning two. So there's essentially two layers of phospholipids. They all pair up next to each other, and then there's essentially a mirror image of them, and it creates a really unique type of chemistry for these phospholipids. Again, so right here I have all of these polar heads lined up with each other along the top. And the same is true down here along the bottom, all of the polar heads, with their then tails pointing and interacting with each other. What's important to remember is that the extracellular space is the space outside of a cell. It is made mostly of water in every living thing. And the same is true for the intracellular space. This is the space inside of the cell. It is also made primarily of water. Again, that's important because the way in which these arrange themselves, the polar heads face in to interact with the water that's on the inside of the cell, and the polar heads face out, again, interacting with the water that's on the outside of the cell. And it forms this nice barrier, this hydrophobic barrier across the middle. This prevents materials that are on the outside of the cell being able to just pass through freely and make it into the cell. And the same is true for the materials on the inside. They cannot just pass freely because they're going to be repelled by this phospholipid bilayer, specifically the lipid portion of it in the center that has the nonpolar bonds. This is what allows a cell to have its own specific environment to allow it to be different than other cell types. Ultimately, what makes one cell type, like a muscle cell, different from a nervous cell is what it contains on the inside of it. And the way in which it can contain those materials is by having a cell membrane. So this highlights and shows that first function of cell membranes, that they act as an effective barrier by keeping the inside materials in and the outside materials out. A good analogy would be to think of this cell membrane as a bubble of oil. Right? If you typically blow a soap bubble, but a bubble of oil would be water would not be able to penetrate through it, which is essentially what a cell membrane is. The membrane of a cell contains water inside of it, which is the contents of the cell, and it keeps the outside contents out as well. A very effective barrier. Let's take a look at some of the parts that make up the cell membrane. This first one here is a protein channel, and there are several of them throughout this cell membrane, but protein channels primarily function in the ability to move materials into and out of a cell. These are key for maintaining homeostasis within the cell. Take a look here at this image and we can see some transport proteins in blue and then the materials that they're transporting in green. You can see the phospholipids in their bilayer there as well. And take note over here, it's less out as the cell membrane as a plasma membrane, just another way of phrasing the cell membrane. Each of these protein channels is responsible for moving one type of material into or out of the cell. And each protein channel is specific to the things that they move. So for example, this protein channel over here might be one that moves only water, whereas this one might be a protein channel that moves only glucose, or as this might be a protein channel that moves only salt. The type and quantity of each type of protein channel will be specific to cell types. A muscle cell, for example, which needs a lot of glucose, will have more glucose channels than, say, a nervous cell will. It just depends on the type of cell that they are and what their needs are. But again, the key here is that these protein channels function in the movement of materials into and out of the cell to control, to regulate homeostasis. There's two basic types of transport that are called passive and active, and we'll look at these in a little bit more detail in a minute. But the three major types of passive transport are just simple diffusion, facilitated diffusion, which is diffusion through a membrane, and then osmosis. Active transport is different because it requires energy use primarily from ATP. Again, here's another cross look again at the cell membrane to look at our next part. Over here highlighted is cholesterol, and you'll notice that the cholesterol is wedged in between the nonpolar tails. You don't find the cholesterol out here in the polar areas of the polar heads or down here as well, that it's sandwiched in between each of the nonpolar tails. And the cholesterol in this case is helping to increase fluidity of the cell membrane, meaning that the parts of the cell membrane are not stuck together. They're not locked in place. And what cholesterol does is it essentially sandwiches itself right in between the nonpolar tails so that they don't stick to each other, allowing the membrane to be a bit more fluid. Another part is this antigen that's sticking out on the outside of the cell. And the antigens are just any type of material that help to identify the cell type. In this case, this one here is a carbohydrate. So this would be the type of thing that you find on, say, for example, on the outside of a red blood cell to identify it as a type A, or a type B, or type AB. And a type O red blood cells, these antigens are just lacking completely, which is what makes a type O. But this is our third function then of cell membranes, and acting as identification would be what we see here with these antigens. In this case specifically, it's a carbohydrate. The cell membrane is referred to as the fluid mosaic model because of this. First of all, to be fluid means that it's something that can flow. It has the ability to move around. Some common things that you know of as fluid are things like liquids or gases. The particles that make them up are constantly moving and flowing and sliding past each other. A mosaic, like this piece of art here, is made of many different pieces put together to make some overall final image. Right? If we take a look back at the fluid mosaic model of a cell membrane, there are many different parts that make it up. There's phospholipids, there's proteins, there's carbohydrates, there's cholesterol, and none of them are locked in place. Like for example, this protein right here could shift and it can move over here depending upon the cell's needs. Every one of these individual phospholipids has the ability to slide past each other and to move on to other locations. All of the components of the cell membrane are movable, and there's many different varieties. That's why it's called the fluid mosaic model. All right, so now let's talk about cell transport and the movement of materials into and out of the cell. And the first branch that we'll look at is what I mentioned before. It would be those of passive transport. All three of these over here on the left-hand side are showing varieties of passive transport. The first is what we just call diffusion. And in diffusion, materials are looking to spread out. They're looking to move from an area of high concentration, so an area where there's a lot of them, to an area of low concentration, to an area where there's not a lot of them. In this case over here, we're looking at oxygen diffusing into the cell. Now, oxygen is so small that it can squeeze right in between the little spaces between the phospholipids. And in this case, on the outside of the cell, there was a lot of oxygen. On the inside of the cell, there was very little. So oxygen will move from an area where there's a lot of it to an area where there's very little of it. Again, think of it as if the oxygen's trying to spread out as equally as possible. So if there's a lot on the outside, it needs to send some of it to the inside. This is passive diffusion or passive transport. It requires no energy for the cell. The cell doesn't have to do anything at all for this to happen. A second type of passive transport is one that's called facilitated diffusion. And these two right here are both examples of facilitated diffusion. In facilitated diffusion, the facilitation portion comes from a protein channel. To facilitate means to help. So in this case, really, diffusion is happening just like it happened over here, but it's just now happening through a protein channel. So right here's a protein channel, and here's a separate one. Now, sodium needs to move through a protein channel because of its positive charge. That charge allows it to not be able to squeeze through the phospholipids like oxygen can. So it needs to move through a protein channel. Glucose is a much larger molecule, so it as well also needs to move through a protein channel to be able to get into the cell. Again, both varieties of facilitated diffusion, again, these also don't require energy. A very common misconception, people see that facilitated, like, oh, well, this must be using energy. No, not the case. Facilitated diffusion does not use energy. This happens all on its own. Also, the other major type of transport is called active transport. Now, this type of transfer does use energy. The cell is actually working to be able to move materials in. This would be the case if you need to move a very large material or if you're going to move something against its concentration gradient. If you take a look at this image here, on the outside of the cell, there's a lot of sodium. On the inside of the cell, there's very little. They're trying to show you that by the difference of font size. The font size is much bigger for the sodium here than it is here. You can also tell by the width of the arrow. The sodium arrow is very narrow down here and it's wider up here. So they're trying to show you that there's not a lot of sodium on the inside of the cell, but there is a lot on the outside, and it's vice versa for the potassium. There's not very much on the outside and there's a lot on the inside. But notice the direction of the flow. Sodium is being pushed out while potassium is being pushed in. That's not the way that they want to go. Remember, they want to spread out. So out here, there's a lot of sodium. What that sodium wants to do is it wants to rush into the cell. So the cell has to work to prevent that from happening. To do that, it uses energy in the form of ATP. It's usable chemical energy to prevent the sodium from coming in and to force more sodium out. The same is true for potassium. The potassium wants to rush out. So what the cell does is it uses energy to prevent that sodium from rushing out and to pull more potassium in this case. This sodium-potassium pump is what it's called. It's a very common active transport. Again, it uses energy. All of these methods of cell transport are key for the cell to be able to maintain its homeostasis so it has the correct amounts of all of its materials. The final type of cell transport is called osmosis, and it's the diffusion of water. And it's so important that it gets its own name. Ultimately, osmosis is just a variety of facilitated diffusion because water does need to pass through a protein channel to get into or out of a cell. In this instance here, we see a plant cell. The one all the way over to the left is in a state called isotonic. It's in an isotonic solution. That means that this cell is like sitting in a water bath, if you will, where the concentrations of everything are the same on the outside and the inside. And so what ends up happening is water will move into the cell, but an equal amount of water will move out. And that's because the concentrations of water are the same on the inside and outside. The water never stops moving because the water particles are constantly moving. As long as the water is not at a temperature of absolute zero, it's always moving. So water will move in, but an equal amount of water will move out. If I took this cell out of this and I put it in a different solution where there was more water in the solution and less water in the cell, the water is going to move from an area where there's a lot of it, which is outside of the cell, to an area where there's not, which would be inside of the cell. This is just like what we saw in our egg lab, where we had water in the vinegar that rushed into the cell, making the egg get bigger. In this scenario over here, a hypertonic solution, water will leave the cell because there's more water inside of the cell than there is in the surrounding environment. And again, in our egg lab, we saw this happening when we put the egg into corn syrup. The water leaves the egg and goes into the corn syrup where there's not quite as much of it. That's called a hypertonic solution. Again, this is a type of passive transport because no energy is expended at all by the cell. It just happens all on its own. So in recap, our cell membranes are used for three things. They're used to act as an effective barrier to keep the inside stuff in. They also act to move materials in and out of the cell to maintain homeostasis. And they also act, and finally, to identify the cell by the antigens.